we've been talking about B cell, and you remember B cell expressed a signal, a receptor when I said MHC. Now let's talk about what is the role of MHC, which is a major histocompatibility complex in immune response. So in this lecture, I will talk about what actually MSC is, and what is the difference between MSC class 1 and class 2. How do we see antigen? How does MSC see an antigen? And then again, what makes an antigen respond, a cell respond to an antigen, and what are the restrictions which are there? And that again will involve a little bit of genetics. It will be always helpful. It will give you a sound understanding, especially immunology, because immunology needs understanding of anatomy, physiology, biochemistry, and cell biology. So this is like prerequisites that we have for you to really have a solid base for that. Now, basic genetics. So this is a chromosome 6, and this is a gene map of human leukocyte antigen. MHC basically was found in mice, initial experiments, but the homologue, the same thing in human, is called human leukocyte antigen, but the word is kind of used for both. It's misnomer, but it's been used for that. So MHC of human being is HLA. So HLA antigen, HLA4, this is chromosome 6 on human leukocyte antigen. We have a long arm, short arm. In the short arm, there's a region which is a HLA region. That's what is important. Now, there are expression of genes. There are expression of genes because this is a chromosome, and you can see that we have class 1, class 2, and class 3. So 3 sets of genes over here, and then there is a subclassification. I want you to pay attention for class 1 and 2 because that's what we're going to discuss in this part. So this is something that you will inherit. Not you will inherit, you would have already inherited. So B, C, E, A, G, F are the choices that you have in class 1. Bunch of choices in class 2 that I'm not interested for this class to know. Then of course I'm very interested for you to know class 2. And these are DP, DM, DQ, and DR. If you ever want to go and donate or receive a organ transplant and they want to do tissue typing on you, this is the report they will give you. They will tell you what genes you have on class 1 and class 2, and they will give you exactly the description of these things. So this, keeping that in mind as a basic concept, let's talk about now what are the major histocompatibility restrictions. Now remember, the whole idea is that cells have to respond to an antigen. Now, if you remember when I was talking about B cell and T cell, I said that B cell can present any antigen and T cell can only present protein antigen. So this basically MSC restriction, the genetic part, is restricted to protein. That's what we call protein restrictions. So your MSC will only come into play when an antigen, like most of the microbes are proteins. They say, somebody said, what is a virus? So one of the definitions of the viruses is they say there's a bad news for you wrapped up in protein. Because there's a message there that's going to cause problem to you, but it's wrapped up in protein. So proteins are there. They are the major components of microbes, and they basically are seen as a threat to the body. And remember, as I said, that when we talked of allergy, immunogens, I also said that as a rule, protein will provoke allergic reaction. Proteins are likely to cause to mount a very severe immune response. Now let's see what happens when a protein antigen is presented. And I already talked about definition of major histocompatibility gene, which was found in mice. The name stays because it's become very common. But the fact of the matter is we're talking about HLA, which is human leukocyte antigens. And these are very tightly regulated, closely linked gene, like the whole HLA part of class 1, class 2, we inherit from our parents, right? And then every vertebrate has MSC having multiple genes. You see, I usually give an example that everything which is written in English, in the books, in the newspaper, in the media, for God knows how many centuries, and they all pick up at least the 23 basic alphabets. So they are these tiny little amino sequence amino acid genes, and then you can combine. So that kind of ability where you can pick up multiple genes give you a unique sequence for each one of us. Now, let's see what actually physically happened, because remember, there are a lot of cartoons, a lot of figures. Those of you who want to have visual, you can see that. I'm going to present to you. And if you want to see the actual one, you may want to look at the electron micrographs for that. This is an interaction between a cell that has just picked up in protein antigen. And this cell needs to present it to those cells which are going to be responsible for initiating, orchestrating, or having a good effector response. That's another term I would use, effector response. This basically is an immune response, and it's more specific. When I say immune cells, so probably I'm talking about many different cells. So immune cell, effector cells, immune response, effector response, for all practical purposes, is the same term. So there is a host cell, like your cell, and let it be a B cell for now. So because we just talked about a B cell. So B cell has got one attached component of a protein, and remember, proteins are larger molecule, they cannot be presented as such, very complex. So they're going to break it down to a very small sequence and a small peptide, and then good enough to be fit in MSC molecule. So MSC molecule basically is just like a mouth. It can just take a limited size of a morsel. So it has a sequence of amino acids. So you have to cut it up, that antigen and protein, and stick it there on top of MSC. And then it's like a groove. So it kind of snugly fit into the groove, and then present to a T cell. And T cell has a corresponding receptor, and then both of them will kind of dock in. So this will be the first step when a B cell meets a T cell, and they dock in those receptors, and then the activity begins. Now, there are different MSC and different T cells. I said that the two important ones that I want you to remember is MSC class 1 and MSC class 2. And they are expressed on our cells. MSC class 1 is expressed on all our nucleated cells. So there's a gene component on all our nucleated cells. And MSC class 2 is expressed on antigen presenting cell. Okay. Now, having said that, remember that when we said antigen presenting cell, I gave you example of antigen presenting cell like a professional, we call professional antigen presenting cell. So this means it's really professional. That is dendritic cell. Dendritic cell. Then there are macrophages and B cell. So these are monocytes as well. These are classical antigen presenting cell. So what it means is that they can only present an antigen if they have MSC on top of them. If they don't have an MSC on top of them, they would not be able to present an antigen. Okay? And at the same time, also remember that these are receptors expressed on the top of surface. And the way I'm presenting, it may seem to you there's only one MSC. No, there can be more than one. There could be multiple. But also remember that we can, and the drugs can, and microbes can upregulate or downregulate expression of these MSC molecules. I repeat, these MSC molecules, which are so important that nothing will get you an immune response, nothing will pass as a signal to make antibodies or any effector respond unless and until it is presented in context with MHC. There are two types of MSC, class 1 and class 2. They are antigen presenting cells that this MSC gets stuck over there, a docking thing, right? Now also remember that we are just presenting a cartoon which shows you only one for description only. There could be more than one. And also remember that they can be either upregulated or downregulated. Okay? Now, and I also said that all this MSC restriction, this is a very common question, MSC restriction is for proteins. MSC restriction is not for anything else because proteins are composed of amino acid peptides and they have to be snugly fit into that before an action begins. Okay. Before I go to the next slide, I'll just pick up two prototype T cells, and you already know them. When I say T cell, it's a broad term, so they express a specific molecule on the top of the surface, which is CD, cluster of differentiation, CD4 and CD8, right? And if you remember that mnemonic, I sent it to you. CD4 was called helper, and CD8 was called killer. Okay? Now, what's happening in this case is that some of the basic information that you may want to write it up and cram it or whatever you want to do. There are two types of presentations. An antigen can be presented in context with class 1, or an antigen can be presented in context of class 2. Right? So let's give an example of class 2, MSC class 2. So, as we already know, this is a dendritic cell, which is an antigen presenting cell. It should have an expression of MSC class 2, which it does, right? And one of the small fragment of peptide is snugly attached in that groove. And this cell, dendritic cell, wants to talk. So the dendritic cell which expresses class 2 can only talk to CD4. I repeat, this is called restriction. So a cell that represents MSC class 2 can only talk to CD4 T cell. And that's what's happening over here. It talks to a CD4 T cell and they come together, they interact, and then there is a response, an immune response. An immune response could be production of antibody, production of cytokine, any activity, any immune activity that these cells can do. So in this case, the example is given of cytokine synthesis. So these are some soluble proteins that are synthesized. Okay. Now, this is a macrophage, and macrophages are also antigen-presenting cell, and they will express, being nucleated cell, they also express class 1. So you see a class 1. So MSC class 1 plus they have a small peptide and they want to interact. Now, as a rule, any cell that expresses class 1 can only talk to CD8 T cell. Okay, so class 2 basically can talk to CD4. So it's like 2 plus 2 is 4. It's easy to, like a mnemonic that you can remember. 2 plus 2 is 4. And 1 talks to 8. And then 8 interacts with that. And then there is an action. And CD8 cell basically, if you remember, is a killer cell. So this cell will kill infected host cell. So there's a message there that this cell has just phagocytosed the bacteria. You said, I've just phagocytosed the bacteria, but its cell is not going to do anything. It's going to break it apart, take a small chunk of protein, put it on top of MSC, present it outside to the cell, invite a CD8 cell to come in, dock with that cell, and kill it. So this is a sequence of events that happen normally when your cells get infected. Okay? Now, having said that, there is, of course, a variability between class 1 and class 2. I already told you they are pleiotropic. We have a lot of choices, so we are all different from one to another. And there are differences, of course, that are genetically different. And the reason is because if you remember that initial gene loci that I gave to you, it was class 1, class 2, class 3. And then again, just like antibody synthesis, VDJ, so if you have a choice, so you can pick one from each, right? And then rearrange them. And this kind of rearrangement where we have different HLA A's and B's and C's from our parents, like father and mother, will give a choice to us that he can mix and match and come up with a new set of cells that are expressing that. That's why we are all unique. The other thing is that I don't want to teach you genetics, but you know for sure that they say that if you want to go for blood donation, you should rather look for your parents or sibling. The chances are that they will be close. Chances are. That happens for tissue typing as well, because of the overlap of one gene with another gene. Not necessarily, but the chances are. Okay? Now, in this case, you can see from here, we call polygenesis. So this means that more genes are there. You can pick up different gene. And there are 6 choices from this group. And there are 3 choices from MSC class 2. So you can have, the whole idea is that there are different expression of gene, different combinations of gene that you can get. That's why we need to do tissue typing. And that exactly happens in our body as well. So polygenesis was one aspect for MSC. We can pick up different gene. The other important concept for MSC is polymorphic. So we may have multiple stable form of MSC that exist in population. Okay? Simple example we call of genetic polymorphism is your groups, ABO groups. This is a sign of your genes picking up those respective. And then again, there are hundreds of slightly different versions we call alleles in a gene that code for MSC. So we have MSC class 1, and there are 100 different genes. The idea is, because there are tons of microbes out there, there are tons of antigen out there, so we want to respond to them. We don't want to respond to them in a random way. We want to be very specific. So immune system specificity is the most important thing. That's why the success of vaccination takes place. And that's what we are going towards. Because I think last year in American Society for Human Genetics conference, and they're talking about human gene, and the whole idea is, we call it immunopharmacology, that they want to give you the drugs according to your genes. So they will do a gene analysis for you, so your drug will work only for you. So that's like, I don't know how many years, they say 10 years. So new drugs which are going to come, they are going to be targeting your gene because we are all unique. So as a rule, all of us should not respond to a drug in the same way because we are all genetically different. Okay? Now, since we have a different choice for picking up different gene components for MSC, likewise, we have a different choice for picking up a particular gene when it comes to BCR as well. Remember, BCR also has that fag fragment which is variable, and then you have heavy chain and light chain. So that kind of a choice, like a fingerprint, is also available. Right? That again comes from the same idea of immune response being very specific. So let's talk about the biochemistry of that. Okay. Now, that much of biochemistry I can tolerate. All right. So A, B, C, and D. If you pay attention to the A1, you will see that this is a cell membrane. We have a cell membrane. And then there are different aspects of that globular proteins that you normally see. So this is a basically, you can see from here, let's go from here, the other way around. So you have a MSC class 1 molecule. So this is the MSC class 1 molecule. You saw in cartoon, now you see the actual biochemical analysis for that. Because by the end of the day, we want to go to that amino acid sequence in detail because of the drugs that will come, either block it or whatever they want to do it. So we have a T cell receptor, and we have a MSC receptor. And this MSC receptor basically, as you can see over here, has two components. And this is a component over here, which is alpha, and there's a component over here, which is beta. And then there's a groove there. Now remember, again, something that you want to remember for good, that this groove can only take a peptide which is 8 to 9 amino acid. So you have a big chunk, you have to cut it up, no less, no more. And just snugly attach it, 8 to 9 amino acid sequence over here. You can see from here, that would then be seen by a T cell. Okay? Now this structure over here, which actually is represented in this one, okay, will be a little bit similar to many different receptors because of the Ig superfamily. Remember when we talked of Ig superfamily, I said T cell receptor, I said B cell receptor, and what was the third thing I said? MHC. So major histocompatibility receptor. So that kind of that. So it has a cytoplasmic tail, and then is a peptide binding groove, alpha 1, alpha 2, alpha 3, right? So these are the different binding sites that we have. And if you look at this one, again, the protein plates and different sequences of carboxy and an amino end. And if you were to look at the sagittal section, you will see over here that this is the groove over here. So there's a beta plate over here. That's where the groove is that you see over here. So that is the groove that you want to target. So if you want to present, if you want to present your cells with a amino acid so that it's processed through MSC class 1, you have to cut it up, make it into 8 to 9 amino acid, and snugly put it over there, and then the T cell will respond. And remember, we sometimes do need T cell response to take care of infection, to take care of cancer, to take care of allergies. So a lot of immunological drugs will be there to modulate T cells. Right? So we need to understand, this is what's called pathophysiology. You want to understand what is going on and what is, to be more specific, immunopathogenesis. So if you see for your microbiology, each and every infection has one of the slides. We say immunopathogenesis. We want to know how is it that that particular microbe causes diseases and how our body fights with it. Okay? Now, the other important thing in this one I want you to pay attention is beta-2 microglobulin. Beta-2 microglobulin, you'll hear that many a times. Beta-2 microglobulin. And it's the same slide, but you can see from here that again, let me come over here, I can't see from here. So you can see pretty much the same. In this case, the previous one was MSC class 1, right? And I said MSC class 1 will talk to CA, right? Now this one is again MSC class 2, right? Again, it's an MSC, and in this case, it has again a peptide binding groove. So there's a groove over there. So this groove can take 12 to 17 amino acids. That was from 8 to 9, so a little bit bigger. And then, again, if you look at the biochemical analysis for that, this is what you will see over here. The other important thing was, as I said, the receptor basically has a outside end, and then a transmembrane end, and then a cytoplasmic tail, which kind of goes into cytoplasm. In this case, because this one over here, MSC class 2, now again it has a peptide groove that you see over here. This molecule, the green one over here, is beta 1, that's what you see over here. And then again, you don't see over here is beta 2, which is a CD4 binding site. So there's a site on MSC which is going to hold on to CD4. So the difference in this case between MSC class 1 and class 2 on the basis of biochemistry is that it does not have beta-2 microglobulin. Rather, it has alpha-2. You notice that? So it may seem insignificant to you right now, but as we move along, you will see what is the advantage of us having or having not. But also remember, they're all proteins. They all are coming from your genes. If there is some deficiency in your genes, you may not be able to make those proteins. And if you don't make those proteins, you will have a problem. Right? A baby is born, and the mother brings the baby like every month, multiple infection. Antibiotics are not working. What can antibiotics do? Because what's happening is that that particular baby has immunodeficiency, and she is lacking beta-2 microglobulin, because that was a requirement for the proper functioning of MSC class 1. Right? So these are some of the challenges that we normally see when we talk of immunodeficiency. So this is the normal immune response, and then there are deficiencies there. Okay? Now, let's see, give you an example, how in real time an antigen comes and what goes on in terms of processing of antigen. So antigen comes and it now has to be processed by the cell. Okay, now this is a, let me give you another term, I'm pretty sure you know that. Antigen usually can come by two routes. They can be exogenous, outside the cell, or endogenous, inside the cell. Okay? Give you an example for this case where exogenous antigen could be a virus protein in a vaccine, just like we give you a flu shot. So you got a flu shot, and you don't probably know what's going on now. The only thing, even a layman will know, I'll get fever, but it will be okay. But how it works, and what is the immunopathogenesis of that, how can we control it, this is your area, your expertise that you should know as pharmacists, as a scientist, as a physician, so on and so forth. Because we need to come up with drugs, we need to come up to understand the mechanisms, If you don't understand the mechanisms, you would not be able to come up with any drug. So you need to go into that detail. Okay, so there is a virus protein. It goes into the cell, and cells basically now want to process that. This cell could be an antigen presenting cell. They are, we call, professional. So you will take the antigen in, right? That happened when you took the virus flu shot. And then you know for sure this is a big protein. And this protein, in order for us to have immune response, has to be cut up and broken apart and presented in a nice smaller residue to your MSC. So there are enzymes. I would not go in detail right now. They will break it apart, and then you have a peptide. You can see from here where these three are broken apart. So these are broken apart, and then again the HLA, for example, the one that are going to make MSC. So also remember as a concept that MSC proteins are there within the cell. The message is there. They are not always hanging out on periphery. Had they been hanging out the periphery, they cannot probably present an antigen. So they have to pick up an antigen from inside the cell and take it out and express it to outside the cell. So what happens is that they have a HLA, which happens to be HLA-D, and there's a groove over there. Okay, there's a groove over there. You see that? And this is blocked. So the sooner the message comes over here that we got an exogenous protein and now we need to present it. And the cell knows we can only present it via MSC, but there's no MSC there. So there's a message that goes, okay, let's prepare an MSC. So what happens is that your cell get the message, ribosomes start making synthesis of MSC class 2 with the chains, and then it becomes assembly, they prepare it, Golgi apparatus comes over here, and then they basically pack it up, all those things that are required like a receptor, and send it to the cytoplasm. So it comes up in a raw form. And the raw form would suggest that while it is in the cell, the groove is there, but we don't want this groove to be occupied by any other thing than the required antigen. So we put a lock on that. So we kind of close it. So we put like a clip there, and it is called clip. So you can see from here, once it's released from Golgi apparatus, we form a clip, and then basically this whole structure will come and fuse with the vesicle that contains an antigen. And then this process, we remove the clip that is degraded and then put on the antigen peptide of our interest on top of that MSC and then present it to the CD4 T cell. So remember, what is happening is why you are being protected by a flu shot. Because they gave you a protein of the virus that's going to cause infection. Your B cells have taken it in. Your cells, according to your genes, have processed it. You picked up your MSC, attach a small portion of that protein, put it up, and then present it to your CD4 T cells. And then CD4 T cells now will prepare whatever is required to have a good immune response. Okay? And this happens with class 2. This happened with CD4. But the other thing I added over here, this happens when the antigen is exogenous, like virus, protein in a vaccine. That's a typical example that I gave to you. Now, you just kind of put that frame, the same sequence of event for class 1. So we presented class 2, we're going to go for class 1. In this case, just kind of switch around the words. So instead of exogenous protein, what do we have now? Endogenous, right? So we will have endogenous antigen. This being the antigen is synthesized within the cell. That's what it means, right? And mostly, many a time, it will be a bacteria taken up by the cell, broken apart, right? big chunk of that. The other thing I want you to remember in this case is that, as I said earlier, every nucleated cell expresses MSC class 1. Only antigen presenting cells like macrophages, B cell, and dendritic cell present class 2. But dendritic cell also have an ability to present an antigen in context with class 1 as well. No, nothing stops them from doing it because they are professional. Does that make sense? Correct? Now, let me tabulate that and then we'll go on. So far, I've talked about characteristics, MSC class 1, MSC class 2, so you can kind of frame that in your mind. If you look at the structure of MSC class 1, remember I told you that both of them have alpha chain, though both of them do have a beta chain. One special thing I want you to remember for class 1 is beta 2 microglobulin. Beta 2 microglobulin. Okay, and when I emphasize something, there's a question there. Many of you will wonder where the questions are coming from. So if I want to ask you what is the difference between structure of class 1, class 2, I want you to know beta 2 in microglobulin, but that is the difference. Now, as far as cellular expression, MSC class 1 is expressed on all the nucleated cells, right? But I also told you, and it may come to your mind, there was only one MSC over there. There could be more than one. It could be as many here as you have. You know, there were tiny little structures. So what we can do it, we can upregulate or downregulate. So when there is a fight between us and virus, guess what? What we are trying to do? What would I do if I want to fight a virus? What would I do in terms of expression of class 1? Should I upregulate or downregulate? Upregulate. But what would virus do? Downregulate. So we need to understand how do we upregulate, and we need to understand how do virus downregulate. This understanding is immunopathogenesis, because you cannot come up with any plan, any device if you don't know how the game is played. That makes sense? Now in this case, one of the important drugs I can give you in action right now, interferons. So if you have hepatitis, they give you gamma interferon. So what is interferon doing? Upregulating MSC class 1 so that more virus gets expressed and more T cells are recruited, and there's a profound, robust immune response. Now, class 2 constitutively on antigen presenting cell, and I've been saying over and over again, and again, this table will just make your life a little bit miserable. Thymic epithelial cells also do that, right? On top of antigen presenting cells, and then they are inducible. So what we mean by inducible means that, again, a background for cancer cell. If I tell you the cancer cell are going to be killed by the cells expressing MSC class 1, right? So our interest is, if we make an anti-cancer therapy or drug, we want to upregulate MSC class 1, correct? And what do the cancers do? Downregulate. So we need to understand how do we upregulate, we need to understand how cancers downregulate. So that's the immunopathogenesis. That was the basis for all drug therapy, that you will try to understand how and why and what happens. We talked about peptide binding groove, 8 to 9 amino acid, we talked about that, 12 to 17. And we talked about binds peptide endogenous. This means something which is catabolized within the cytoplasm as compared to exogenous. The only exception to this one was cross presentation. I'm going to discuss in a while. I just gave you an idea that dendritic cells have an ability to cross represent. So they should present by class 2, but they have an ability to present class 1 as well. And we already know the rule. And we're going to add, we start with the basic thing, and we get it getting complex. So you have to have the basis and then add ons. So I said class 1 can only talk to CD8, and class 2 can only talk to CD4, and then we just added NK cell. The reason being that no science in medicine has advanced with such a speed as immunology, to such an extent that I myself feel guilty to teach the textbook of Koiko and Sunshine, which was published in 2009, because things change within years and months. That's why I was talking to our group of people, ASM. Graduate education basically is research. What makes you different or makes anybody else studying in any other school, the difference, not the textbook, because they have the same textbook, not the classes, we have the same classes, they're all same thing. Difference is research. Because you have an ability to think, you have an ability to process information, and you have an ability to critically analyze the thing. So that is the major thing. So things keep on changing. So don't think, but again, we have to start with the basis. We have to start with the basis. Okay. Now, selective binding of a process peptide by different MSC molecules. So you can see from here, if this is a protein antigen, a little bit of detail of what we already said. So proteins is very complex, primary structures, secondary, tertiary, and no, remember, this is an important slide for you to understand why is it not that they are able to make good vaccine, and why is the vaccine not working, and things of that sort. Think. Now, this is a protein, a viral protein. It has no less than 122 sequence of amino acid. And we have enzymes over there. They will break it up into smaller chunks. And you can see we process these peptides, and then we basically add it up on top of your MHC. Okay? So bigger, larger protein cut in different pieces. But remember, many things I'm not talking right now. I'm not talking about the enzyme. I'm not talking about the rate. I'm not talking about what causes your cell to pick up one particular peptide and put it on top of your MSC and not the other one. Because that is an area of research, and we don't want to go over there, so we want to just make things simple for you. And this is another slide where in the previous one, you remember I said exogenous antigen, there was a viral protein or vaccine. Now let's see actual virus. So there is an actual virus infection, and viruses basically cannot enter your body unless there's a receptor there. They bind to the receptor, they go over there, there's a synthesis of viral protein, and there is a antigen which is prepared within the cytoplasm. We call it endogenous antigen. And again, cell biology, there's a machinery in the cell. Again, the message will come that we have just received a virus and we want to prepare an MSC. So what do you think the message will say to the nucleus? What kind of MSC do we need to prepare to handle a virus? MSC class 1 or 2? 1. And the cell knows it. So it prepares, it assembles, message goes to all these transcription, translation. We're not going to go to that steps. Ribosome, and then it starts synthesis of class 1. The important thing at this stage is now that it wants to make sure that it adds beta-2 microglobulin. Because you have to now be extra careful as compared to MSC class 2, because we have to have that for a proper functioning receptor. And then there is assembly of class 1, and there beta-2 is very protective. You want to chaperone it, make sure it gets over there. We attach everything, and then we basically, important thing in this case, you see the difference between class 1 and class 2 is that we are not processing it into a acid vesicle. In the class 2, we had 2 vesicles fused with each other. One containing the antigen, the other containing the MSC. Bring them together, open clip up, add on top of it, and present it to the T cells. In this case, what's happening is we are basically bringing the peptide into the nucleus, right? And there where is attachment of that peptide takes place in the groove, and then the whole structure goes into the Golgi apparatus and then basically hangs out and is attached and presented to CD8 cell. So this process, I'm just going to use some immunological term, this whole process is called signaling. And this is, since this is a basic immunology class, I don't want to go in detail, but each and every step within the cytoplasm in the nucleus has tons of enzymes, has tons of other players over there. But if you understand the basis for that, it will be easy for you to go from there. Okay? Now, just to summarize what I said, there is a virus-infected cell, right? That's endogenous thing. And this is broken up into different pieces, right? Dendritic cell has a capability. It should do it to MSC class 2, but it's very clever. It knows that viruses are going to be dealt with CD8, mostly. So it wants to present it in context with MSC class 1, CD8 comes, and we have virus-specific CD8 T cell. This process over here, where a dendritic cell takes up an exogenous antigen, for example, derived from a virus-infected cell, and process the peptide with class 1 inside the cell and presents it to a CD8 cell. We call it cross presentation. So this is a cross presentation. Now the question will come, what happens if our cells are unable to present an antigen? That will be a disaster. That's where the problems come. Now, theoretically, it's easy, just like common sense. What makes your cell unable or unable to respond to an antigen? Number one, remember, so far we have discussed that antigen processing cannot occur unless and until you have class 1 and class 2 presentation. Agree with me? Because the final major players for immune response are CD4 T cells and CD8 T cells. They would never, I usually say that these are blind. They would never ever see an antigen unless and until it is presented in context with MSC class 1 and MSC class 2. Okay? Now, things are wrong. You have a problem. There's a disease. There is an immunodeficiency. Immunodeficiency, you can have congenital, or you can AIDS patient. You can acquire as well. Immunodeficiency. Right? There are many aspects for that. You can follow infection, radiation exposure, tons of things. But let's talk about that you are unable to respond because you have limited MSC class 1 and 2 per person. So your machinery of making class 1 and 2 is limited. And then the idea is that your cell should generate at least one peptide. Remember, that's kind of a minimum that you have to generate within the cell to present to the T cell. If they are even not presenting one, you make a vaccine and it doesn't work. Why? Because your cells have not even picked up one component of that virus and presented it to the T cell, even one. So that's ineffective, right? The idea is good vaccine should recruit more, right? And that depends upon your robust immune system. So technically, if entire antigen fails to generate even one peptide, no response, right? And then the other problem will be naturally occurring pathogens. They are very clever, right? We'll talk about that in compensal bacteria, but some of the bacteria fool our immune system. They disguise. They come with a cover. The immune system is unable to recognize them. And finally, you are unresponsive. You're taking up the antigen because you cannot basically synthesize polymers of amino acid, and especially in the case when there's a vaccine with a single small peptide. You prepare a vaccine which just has small peptide, the chances are it's not going to be good vaccine because you're going to hit or miss because of the size of that. Okay? Now, if you pay attention to the whole process, now so far we have covered the context of MSC, the antigen comes presented in context of MSC class 1 or 2, and I said the T cell comes, CD4 or CD8, sees the antigen, then I said it has to be an immune response. Now the question is now, what is an immune response? Or let's talk about a little bit of anomalies, what can go wrong, and what can bypass this system. Like, for example, electrical conduction. So what can short circuit it? Maybe that's a better word, short circuit. Now, you can see from here, other type of antigen activated T cell responses could happen, we'll discuss in a figure, when they are super antigens. So that's another term. Some of the antigens are super antigen, so they kind of short circuit your system. They bypass that. And remember, by rule, these are dangerous because they're going to play havoc with your immune system. Number one, bacterial exotoxin. Staph enterotoxin specifically. And the other thing is that any antigen which is not processed, and basically any intact molecule that bind with MSC. Remember I told you that in order for you to have immune response, normally you should fit that peptide into the groove of MSC, right? But these are cell-to-cell signaling. You can bypass that when some of the antigen will come and attach to the side of MSC and may be short circuit that, okay? Or this is a very, I would say, weak response that you call in help from CD4 T cell, and there are tons of them, but for some reason, this whole process is able to activate only 10% of total CD4 T cells. So you have to recruit a lot of CD4 T cell for a good immune response. So that could be another thing. And then again, some of the conventional antigens, because they basically have fewer clones, and the typical example is the peptides basically presented with MSC. I'll talk about that in detail. And finally, one of the clinical nightmare will be toxic shock syndrome. Toxic shock syndrome, again, there is a super antigen and it's going to cause shock. Because remember, if you are going to stimulate, it looks very simple to you, if you are going to stimulate that system, you will go into shock. The whole body, the whole immune system is going to go into shock, and it will be very difficult to take control of that. And the good example for that is, and you can see from here, A and B. Number A, then again, is an interaction between a cell that is expressing MSC class 2, and there's a TCR region, and you remember there has to be a peptide over here, right? Now, in this case, what happened is, I said, the super antigen binds outside MSC class 2 and interacts with it like a short circuit. So you have a super antigen which will kind of bring them together, make fool of the system, and the system will explode. When that happens, that is a very serious consequence, and that can happen in many different infections, and we talked about that, and we will talk in detail. The other important thing in this case, again, is a MSC molecule with beta-2 microglobulin, as you can see from here. So which kind of MSC is this, if it has beta-2 microglobulin? Class 1 or 2? There you go. So you have a 50% chance of giving a correct answer, and you make a mistake there. All right, so then there's a TCR. So which cell is coming and talking to MSC class 1 over here? Which cell will have that TCR? The TCR belongs to which cell? Huh? Why are you so reluctant? T cell. Which type of T cell? MSC class 1 will only talk to CD8. Does that make sense? So this TCR belongs to CD8. And this TCR belongs to what? CD4. Because this is the normal relationship that these people have. All right. So there is a glycolipid antigen. There's a glycolipid antigen, and then it again causes that short circuit, and then will have the system explode. Okay? Yes, no? Okay. I was talking to somebody, so sometime in immunity we have to give the booster dose. So remember, for you it's a childhood vaccination. You bring the child, and the child sees the needle, and the child is scared. Don't inject me, and they're going to hold on to the child, punch in. That's what probably is happening right now. Call you up at 8 in the morning, you're not interested, and say, why on earth is he calling me? So there's a shot. Okay, but it's going to be very protective, and it's going to be helpful, believe you me. All right. We talked about polymorphic MSC class 1 and 2 gene and said that there are a lot of choices that you may have. And then again, let's talk of a little bit, not much, of polymorphic MSC. And after this, I'll have like 10 minutes break, and then we'll continue. And this is actually the simplified human histocompatibility complex, which is called HLA. So again, HLA class 1, 3 choices. You have HLA class 2, you have these genes, and then again, your body will pick it up. You can see from here, and then these are the sequences that if you today decide that I'm going to go and get my tissue typing, so they will give you tissue typing, and they will give you your number for class 2 and class 1. They're going to give you CQ for DP, DQ and DR, and ABC. So this is something that they will give you in detail, and that's what we normally do. Now, these are basically inherited genes, so you inherit them. And this is a gene kind of a map that you can see, and this basically will give you an idea as to where are these coming from. So you can see that it is coming from your parents. So there are different, at least 4 different HLA haplotypes, father 1 and 2, and then different haplotype, mother 3 and 4, and these are the 4 kids that they have. And then you can see over here, the children of these parents will express 4 different haplotypes. So it's going to be one of the 3 or one of the 4. And you can see they will all come with unique set of haplotypes and set of genes which may resemble a little bit, but if you pay attention, they basically are unique. So that is where it is kind of coming, and that's where diversity is coming. The question that comes, why do we need to have diversity? Why do we need to have that? Now, homeostatically, remember, this is a species protective response. This is a protective response that we have, and we don't want to blame our parents for that, but that's what it is. Because you cannot go back and change the gene, right? But of course, protective. Then again, it is associated with susceptibility or resistance to infection agent. That's another thing. This is coming from that genetic background. And then again, some of the things, like in science, we don't know. There may be some other mechanisms that as of today, we are not familiar with. Why is it that all of us carry a different gene, and what's going on in terms of the genetic thing that we have? Okay? So I'll stop here, give you 10 minutes.